All right. Good morning, everybody. Today is Friday, November 11th, 2016. And this is Before the First Cup with Jules here at UCY.TV. Happy Friday, everyone. We made it. This was probably one of the longest weeks yet. It is 45 degrees and really cloudy and leafy here in upstate New York. I opened my back door to run out and get my refill of coffee that I get daily before the show. And there was a mountain of leaves outside my door. So where do we leave off yesterday? We titled the show Precision Medicine, DARPA, DNA Factories, Biopharma, and the election. We took a look at how stocks, in particular, those stocks related to biopharma and some of this mad science technology did after the election. They had been crying, oh, the Dow was down. Oh, world meltdown. But you know what? We had biotech stocks go crazy. And everyone's saying that this is going to be a big economic boom. We have looked at how Obama has passed all kinds of executive orders and edicts supporting 21st century cures and 21st century technology, connecting us all together to the machine, eventually to open your arms and accept the machine. We can't have resistant consumers out there. And they've had a lot of trouble trying to convince the other side that having technology in your life 24/7 is a good idea. You know, there's lots of Luddites out there still. Typically, we're older. Typically, we've looked at this as well. We are the generation that stood on that cusp of before and after. And we now have been handed the baton as to how the future is going to move forward. And I hate to tell you, but it doesn't look like the changing of the guard is going to stop anything. These dominoes have all been lined up. This is all going to be done in the name of saving humanity, curing cancer, making you be healthier and live longer, which will, of course, reduce the cost of health insurance and your carbon footprint on planet Earth. This is how it's going to be sold. This is how it's going to be pushed. And now we have this step forward into a more personal position than we've ever seen technology before. This 21st Century Cures Act that's sitting out there on the floor. We listened to that advertisement video yesterday. I can't, every time I hear it with that music, it just drives me crazy. I don't know, it's in the notes from yesterday's show. But that 21st Century Cures Act is still waiting to be debated. And we know that after this election, that the Republicans own everything. They now have the White House. They have the House and the Senate. They're going to have the Supreme Court. And so we better just hope that they have our best interests in mind. And I have a very bad feeling, just by looking at all the signs, we took a big, long look at DARPA over the last few days. At the history going back to 1997 and even way before. I mean, we looked back at Dr. Arnston. I mentioned him yesterday, the day before. I linked to his edible vaccines and all of his papers and stuff. But we did some in-depth study on that a while back. Just search Before the First Cup and edible vaccines. You'll see there are probably 2 or 3 shows on that. That all ties in with genetic modification and being able to even splice and dice drugs. And now, with some of the research they've been doing, they've been able to program cells, bacteria, and viruses to actually be the carriers of the genetically modified material that they want to get into other living organisms that could then potentially get into us. We looked at DARPA's Insect Allies program a couple times now this week. where they're going to infect insects with genetically modified organisms, whether it be viruses or bacteria. I don't know. There isn't tons of detail yet because they are just looking for proposers 1 week from today. It's going to be the proposers meeting. And those insects will then be released on crops, and they're talking about any and every type of crop, to be able in one season, as they're munching on those plants, infect the plants with a pathogen that they are directing through their modification. I will guarantee you there will not need to be any type of labeling on that, because they went all out and said how they're using a natural process to get into these plants. That's kind of the key. Even the CRISPRing, we found out those CRISPRed mushrooms, they don't have to label as GMO either, even though technically they are genetically modified because they went in and flipped some switches. But they aren't genetically modified by the traditional sense, because in the old days, they were actually inserting foreign organisms into the food that then would go in and make changes. Well, now they're just flipping switches in the organism to be able to do that. And, as we've been now looking more and more into some of these insane programs that started at the National Institute of Health and kind of spun out, and DARPA then even spun it out to its own whole division called the Biological Technologies Office. We found that they've been doing tons and tons of sprinkle research funding on how to create living factories, whether it be by utilizing biological material or synthetic material, right? They've created 2 new synthetic proteins, X and Y. And like I always point out, it's not male and female. They just made it sound like something that was familiar to you. No, these are actual proteins, CATG, and they made 2 more, X and Y, to add to the double helix that will give them exponential amounts of godly creation powers to create life forms that don't exist anywhere in the universe. We read yesterday, I don't know if we read actually any of the story. I might have just hit the headline. There's a story out of Nature magazine from this year, March 17th, 2016. The living factories of the future. Scientists are designing cells. And these are cells themselves. These are living organism cells. They can program cells with a language now. They can push little buttons just like they were writing code, but they can be writing cells. Scientists are designing cells that can manufacture. So this is where we kind of ended yesterday, talking about the factories. I think that was in the title. Yes, the DNA factories, but they should have been called living factories, because it's not just DNA. I mean, we looked at DNA yesterday with the, I don't remember the name, the place that Georgie Porgy Church is over at the MIT and Harvard group. They got a big fat chunk of change over there to start creating a DNA factory, pumping it out to DARPA's needs. But these were actually talking about factories with all kinds of things, not just DNA. Scientists are designing cells that can manufacture drugs, of course. Ding, ding. Reason why biopharma tech stocks are going crazy. Foods as well. And just taking a look at that Insect Allies program. Some of the things that they can manufacture in cells, put into bugs, and set onto crops. That would make a lot of sense, as well as materials. We did look at the DARPA Living Material Program, houses that will literally be made of biological cells so they can sense the environment around them and be able to react in real time. And even designing materials, drugs, and foods that can act as diagnostic biosensors, and we have talked about that also. We will have living little DNA nanobody things in our blood that'll be able to react real time to whether or not your body is operating within parameters. But first, they must agree on a set of engineering tools. From an evolutionary perspective, yeast has no business producing a painkiller. Yeast is one of those organisms they've been doing all kinds of crazy stuff with, yeast and E. coli. But by re-engineering the microbe's genome, Christina Smolke at Stanford University in California has made it do precisely that. Smolke and her team turned yeast into a biofactory that, by starting with sugar as a raw ingredient, makes a potent pain relief drug, hydrocodone. Oh, what? Oh my God, they can put hydrocodone in your beer or your bread. The feed is a prime example of synthetic biology in which scientists reprogram cells to replicate products found in nature or even make more specialized materials that would never normally be produced by a natural organism. Guys, we literally just talking about Trump discussing bringing manufacturing back to America, except I hate to tell you, we listened to Eric Drexler about nano factories and starting from the atomic level and creating from small up. That is going to be the future of manufacturing. It is no longer going to be steel girders. It is going to be getting iron atoms and oxygen and putting all those little pieces together and then 3D printing them into on-demand shapes and requirements. It reduces inventory. It reduces the required number of people to work. Ding. It reduces pollution. Remember, I told you I actually mentioned Bethlehem Steel burning yesterday, a few days ago, just outside of Buffalo. One of their big old buildings. It was really disgusting. It was black smoke. And I just cannot imagine the devastating amount of toxic particles that were in that smoke. I told you when I was a kid, we would drive through Lackawanna. It would smell like a big, nasty, sulfur, horrifying chemical fart. And all the houses literally were orange. It didn't matter what color the siding was. The houses were just coated in orange. Imagine what our lungs looked like breathing that crap. So they're not going to allow, they've already passed a lot of these climate change agreements that won in Paris last year. Regardless of whether or not we're going to pull out of something like that, I don't think the world is going to look too favorably. Even China has agreed to back off, and they are the worst polluting country on the planet. I think the future is going to be sold as something sustainable, quote unquote, 21st century. Agenda 21, save the planet, greenies. It's all going to be done in the name of profit. Synthetic biology is off the charts. That is the 21st century. Synthetic biologists are ambitious. Quote, here we go. We'd all love to imagine a world where we could adapt biology to manufacture any product renewably, ding, quickly and on demand. What did I tell you? This is the way it is going. Trump is not going to save us from being remade in somebody else's image. Groups around the world, and that was the other thing, this has all been democratized, are engineering yeast, bacteria, such as E. coli, and other cells to make plastics, biofuels, medicines, and even textiles with the goal of creating living factories that are cheaper, simpler, and more sustainable ding-a-ling, than their industrial counterparts. For instance, and remember guys, China's way ahead of us in this biotech game. The biomaterials company Speber Inc. in Tushikura, Japan, has reprogrammed bacteria to turn out spider silk for use in strong, lightweight winter clothing. But synthetic biologists, remember the spider goat? They were turning out spider silk way back when. I don't know, it was 2009, 2011 maybe. Squirting it out in goat's teats. Well, now they don't need that anymore. They just have bacteria spitting it out. But synthetic biologists are going beyond simple producing materials. They are creating complex systems by wiring up genetic parts into circuits. Ding! Merging of biology and technology. Guys, this is coming. No matter whether we want it or not, I'm telling you, they're going to get it into us. I'm going to show you how we can get there eventually today. It's all going to be in the name of the new medical establishment. And, these prices, I was telling you, just from a personal perspective, health insurance prices have gone off the charts, freaking insane. We are looking at, I just saw something this morning about all the different plans, the bronze, the silver, the gold, the platinum. And this is in general across the board, across America. People in general are saying they can't afford more than $100 a month for health insurance. And if we're talking $100 a month gets you maybe, and I don't even know what it would get you. Let's even just say it got you $50 prescriptions and maybe a couple of doctor visits a year for free. And after that, you're going to have to pay through the nose plus $100,000 deductible. What is that going to get you? If the platinum plan is $600 a month and a $13,000 deductible, this is unsustainable. And even if it gets overturned, even if Obamacare gets flipped on its head, there's got to be a solution. Well, what is that solution going to be? Well, it's going to be some new plan. I really, really, really hope it is free market across all state lines and make all those big insurance companies compete for their business and offer value. But I have a bad feeling that part of the cures that they are going to offer as a part of those potential losses they may take by not having a monopoly anymore is going to be to fully reduce costs, fully automate healthcare, telemedicine everywhere, and remote biosensing and monitoring to make sure you're operating within parameters, to set guidelines. You need to quit smoking. You need to lose 20 pounds, statistically speaking, for your age and height. Johnny, you're a little bit overweight there. And now we can even tell through your analysis of what's passing through your small intestine with our new, what was the name of that company? Synthorax, or I don't know, Bill and Melinda Gates had that investment in those new living therapeutics. Synlogics, that's what it was. The new reprogrammed gut bacteria. We've been able to determine that you've been eating too many Cheetos. Johnny, not good for your, I don't know, what would Cheetos affect? Your blood sugar, maybe? I don't know. You can't eat anymore, and if you do, we're going to have to take some drastic measures. We're going to have to reprogram you. We're going to have to release the synthetic bacteria in your gut that pushes those little buttons that makes those cravings for that high fructose corn syrup go away. I mean, guys, this is all within the realm of possibility. And I just think that this is going to be the solution to curing our quote-unquote health insurance woes. But synthetic biologists are going beyond simply producing materials. They're creating complex systems by wiring up genetic parts into circuits. This approach has already resulted in various living switches and sophisticated sensors. For example, Martin Frussengerger's group at the Swiss Federal Institute of Technology in Zurich has built biomedical sensors, that can detect disease-relevant metabolites in the blood and trigger the production of therapeutic compounds. There we go, guys. Little nanobody thingies, whatever they are, little sensors that they've programmed like circuits in some sort of little living thing that they put into your bloodstream, and it actually, when you went outside of parameters, started producing quote unquote therapeutic compounds. We better trust in them that those therapeutic compounds aren't going to be making changes that are off target, right? We found out a lot of the stuff, including CRISPR, has off target effects. In mice, of course, these biosensors successfully staved off gout and obesity, as well as treated the skin disease psoriasis. And you can see another story on this called Living Pills. Pills are alive. They're alive. This young field has already spawned some success stories. And like I said, they are treating all this stuff just like technology companies used to treat the way that we would sell new networking technologies. We would write a success story, put it into a big glossy, and then take it to potential customers and say, look, look, look. We had these case studies. We almost gave the product away for free. Step right up, be our guinea pig. And when it went well, we used them to advertise and sell it to the masses. But they say making and putting together genetic parts currently involves substantial guessworking and unpredictability. Well, who would have thunk it? So the only people that are going to be getting this, right, we have established this, are the desperate, the ones who have one more chance. Step right up for your phase 1 clinical trial and be our human experimentation, something that was not considered ethical at the beginning of this year, but is now becoming modus operandi. For this field to advance, academics and industrial players must agree on a toolbox of reliable genetic parts and the best strategies for assembling them, and be certain that all this information will go into the cloud and we will democratize it more. To build an artificial product, synthetic biologists begin by selecting DNA parts on a computer, and then they manufacture them with specialized instruments. And then the parts can be inserted into a DNA of a microorganism, and as well as cells, to reprogram them. And thanks to the plummeting cost of DNA sequencing, right, we found out the other day, $29.95 on sale. You know, I just actually saw, too, a second ad advertising for Christmas. How great. Get it all for your family. You'll get a discount if you buy 5 of them through Discover Magazine as well for DNA toolkits as a gift. Thanks to the plummeting cost of DNA sequencing, there is now a vast collection of genetic data through which synthetic biologists can sift to find useful genes. And yes, that was formerly called the Cohort Program, now the All of Us Program. Their goal to get 1 million human beings to submit DNA to an open database for research. Quote, biology has given us this big, crazy library of stuff to choose from, and DARPA had even talked about pushing the buttons, creating life forms on the fly. Well, that is exactly what we are doing. And that was a quote from Christopher Voigt, a synthetic biologist at MIT and Cambridge. One leading database, the U.S. National Center for Biotechnology Information Gene Bank, contains already 190 million DNA sequences from over 100,000 organisms, not just humans. Some of the most widely used genetic parts encode enzymes, which are the proteins that are essential for manufacturing. To transform glucose into hydrocodone, for example, Smokey's team took 23 enzyme encoding genes from diverse species, transgenic, and then they put them into a yeast. The Tower of Babel is here. Our favorites in the genetic designer's palette are promoters. These are stretches of DNA that regulate the activity of nearby genes and then cause them to be expressed. When proteins called transcription factors bind to a promoter, the process of transcribing a gene begins. And, you know, transcribing genes is one thing, but making a mistake and trying to get them back is another. We read yesterday that DARPA has a new program out there called Safe Genes that is looking at a way to put digital rights management on our living genome so that if we ever accidentally or on purpose overwrite our genetic code, whoops, we can do a rollback and get back to where we were before at a timestamp. Pretty frightening thought, especially considering now that genetic modification, CRISPR, and these types of technologies are considered WMDs. That is the whole reason that I predict that this is not ending. They have invested so much time, effort, and money and democratized this so far across the planet that there's no stopping this bus. And anything that the new president is going to have to deal with will all be about protecting us, keeping us safe from biowarfare, whether it be through infection or through agricultural. That is another way that they attack populations in war, all in the name of warfare and curing disease. So, I don't know, cellular software, thanks to greater automation, thank goodness for automation. It is now simpler and cheaper than ever before to make synthetic DNA parts. But connecting those parts to form a genetic circuit that can work together to provide sophisticated computer-like behavior has still been a challenge. Anytime you physically connect DNA, you are connecting a new sequence that can interface. As DNA is so information rich, you can create a new promoter or change the beginning of an RNA. And even carefully designed circuits can malfunction, ding, and cause unwanted expression of a gene, oh no, or interference between other genetic elements in that biological circuit, and outcomes that cannot be foreseen in computer models. The community is very much operating in a world where we cannot predict what is going to happen, oh my goodness, in our system as we build them. So all the more reason to just democratize this across the planet and let every lunatic in a lab start creating life forms. I mean, are you kidding me? How can we be sustainable? This would tell me this is exactly the reason why a lot of the tech billionaires are now putting their time, money, and effort into getting off this rock, because the end result of this does not look pretty. This uncertainty means that many of the steps in engineering a synthetic system need to be tested and optimized, and software tools and robotics, ding ding, are speeding up each part of this process, from building the artificial DNA to inserting it into a microbe. So, let's now get AI such as Watson involved. Watson is being trained in genomics. And let's make certain that artificial intelligence in the future is the one that's creating artificial life forms. I mean, that literally is what they are talking about here. You can use high throughput prototyping to just build every variant, and hopefully one of them will hit. The push for automation has led to a number of synthetic biology research centers and firms to install BioFoundry facilities in which robotic assembly lines create, test, and optimize microbes at a much larger scale that can be done by hand. BioFoundries are enabling synthetic biologists to embark upon ambitious projects. For example, Voight, who co-directs the MIT Broad Foundry, and that is the one that got the $32 million grant from DARPA to do just this, cites a collaboration with the Swiss pharmaceutical company Novartis to manufacture a huge range of molecules that are produced by bacteria in the human gut. Ding, ding, ding. Here we go. This is getting it into our bodies. Other institutions pursuing foundry models include Synbesite program in Imperial College London. Oh, that's where Oxitec came out of, as well as National University at Singapore's Synthetic Biology Foundry. Also, the U.S. Defense Advanced Research Project Agency, ding ding, more affectionately known as DARPA, has also invested heavily in the MIT Broad facility. We talked about that one yesterday, including a 5-year, this was in 2015, $32 million contract that began October 2015, so that takes us up to 2020. Some biologists remain skeptical that the rush to scale up and automate and favor a more theory-driven strategy. The perfect host. Well, species that are commonly used as model organisms in the lab, such as brewer's yeast, like I said, therapeutic, craft beer, as well as the bacterium Escherichia coli, more affectionately known as E. coli, like we said, have also been pressed into service by synthetic biologists, and many breakthroughs in biosynthesis have been achieved with these organisms, such as when Kiesling and his collaborators in Amaris, a company that he co-founded in 2003, reprogrammed S. cerevisiae to manufacture the anti-malarial compound artemisium. But these common lab organisms are not necessarily suited to be grown on an industrial scale, so the hunt for better alternatives has led scientists to search in obscure places. Well, that's got to be a little frightening. In some cases, the choice organisms will be those that can withstand harsh manufacturing conditions, so hard to kill, in other words. Maybe you're producing something that's toxic, but volatile. So if you have an organism that can produce it at relatively high temperatures, you could boil it off while you're producing it. Scientists are also testing whether it is possible to feed microbes with what? With carbon sources other than sugars to make products. Guys, we talked about another DARPA project a while ago called ITER, if you remember, and that was all in line with some of this autonomy AI that is coming. Another thing, by the way, getting back to autonomy and AI from a few weeks back, and Trump in the election, we discussed the whole wall situation and whether or not that's going to happen. There isn't going to be a physical wall. I'm going to tell you right now, it's not going to happen. What's going to happen, because that one SquadX PDF directly off of DARPA.mil talked about it, being able to patrol our borders using autonomous robots, whether they be in the air or on the ground, and communicating real time with not only HD cameras, but also with lasers and weaponry of some sort, facial recognition, and facial determination as to whether or not you are friend or foe based upon the genetic traits that are viewable in your face and compared against a database. They'll be able to compare you against a criminal database as well. That's easy peasy. License plate scanners. And they might even have robotic arms and lasers, grenade launchers. What else did we see? Tasers. Maybe some non-lethal weaponry. And then put out the call, whoop, 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 to the nearest border patrol. And maybe in the future, they will even be Valkyrie robots. Those same Valkyrie robots from NASA. NASA's got their new robotic challenge. We looked at that as well. Originally, those robots were being demoed in the DARPA Grand Challenge. The DARPA Robotics Challenge, Grand Challenge was the shape-shifting Internet of Things software. But in the DARPA Robotics Challenge, Valkyrie was going to be a search and rescue robot to respond in the name of a disaster. Now they are on phase 2 of that. There is a NASA Robotics Challenge, and they are going to take Valkyrie and make a next generation to do things such as aid on the space station for repairs, as well as to potentially head to Mars, where they will be trained to build structures and power infrastructure. They've also suggested that those very same Valkyrie robots, phase 2, might also be utilized to aid in natural disasters and man-made disasters here on planet Earth, to be able to dispatch en masse to go to an aid area and help rebuild. Well, how much of a stretch would it be to take another one of these armies of robots, maybe even an Atlas version too, we've seen that as well, and make those available when your surveillance apparatus of autonomous AI that's scanning the border puts out the call for someone to come apprehend a captured individual. So, I don't know, we got a little off the topic there, but I'm telling you, none of this technology that we've been talking about on this show is going to end. They're going to use the situation that we are in to further this technology. And now we are talking, so I mentioned Eater. Eater was a ground robot that they were developing. This was already quite a while ago. I don't remember the year. If it was 2008, maybe they started talking about this. And then we also read about a project that used flies, actual meat, to power the robot. The Eater robot could use all kinds of things, traditional and non-traditional fuels, all the way down to potentially human bodies. Well, now we are talking about them creating microbes that they could feed carbon sources other than sugars to have it turn into a biofactory. Guys, this is a pretty frightening thing. Accidentally or on purpose, if you've been deemed one of those people that is just not ever going to fit into the mold that they want for 21st century world here, who's to say that they couldn't genetically modify a special crop that is somehow given to people just like you? And maybe people just like you, statistically speaking, all do one thing, or eat a certain thing, or drink a certain thing, or smoke a certain thing, that they could just taint a whole product line in the future, or a medication, or a pill, or a vitamin. And it's just crazy. It is crazy. There's going to be no way for us to know what is going into anything anymore, or whether it is natural 1.0 or synthetic 2.0, or whether it's been tinkered or tampered with at a genetic level. Synthetic biology company Exec, and we have talked about these guys a bunch of times before, in Germantown, Maryland. What a fitting place for them to be, is working with bacteria that feed on methane, a cheaper and more efficient means for producing carbon-based products than sugar. When it comes to medical applications, synthetic biologists are engineering mammalian cells, ding, rather than microbes. Such designer cells could produce drugs in response to disease, or take over, ding, 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 certain physiological tasks in people with metabolic disorders such as diabetes, or anything else that they deem to be a disorder, I say. But engineering mammalian cells introduces a whole new set of challenges. All the tools that we have in yeast are just not there in mammalian cells. We don't have as many promoters or tools for regulation of gene expression or protein modification, but don't worry about that because they're already working on it. And maybe we'll get there today or on Monday. And this I thought was quite surprising and made me raise an eyebrow. Just not only what they're talking about here, but also the words they use. The easiest cells for them to cultivate for medical applications utilizing mammalian cell lines are to cultivate tumor-like, ding, especially since they've been doing all this research into curing cancer with the stuff, tumor-like, immortalized, ding, 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 cell lines, which are inherently, ding, 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 defective and therefore not representative of healthy tissues. Now, I'm going to read that sentence with no dings again, but I'm going to tell you that there is something extremely nefarious afoot here, considering the mass amount of research that has been done in synthetic biology, getting it into our bodies, and curing, quote unquote, cancer. All right, let's read this sentence again. No dings. The easiest cells to cultivate are tumor-like, immortalized cell lines, which are inherently, quote unquote, defective, and therefore not representative of healthy tissues. Conversely, tissue-derived primary cells are hard to cultivate and to manipulate. And that is the end game here, people. Manipulation of biological processes. And differences between cell types confound efforts to build toolkits that can be applied across the body. Something that works in a kidney cell will not necessarily work in the lung or the liver. But to get around this, and we talked about this before, they use these Trojan horse technologies. Something that, to get around this, the team is now engineering prosthetic gene circuits, which are introduced into a host cell that can then be implanted at the disease site. Tinkering with genomes can present problems. Even quote unquote smart genome editing tools such as CRISPR-Cas9 can have unpredictable outcomes. I've said that a bunch of times too, and they're like, oh yeah, CRISPR, let's just go forward with CRISPR, send it all out around the planet, add a gene, democratize it all. But whoops, we made a little mistake there, Jenny. I mean, it's so unbelievable that we are at this point, and there really are no controls on this. There are teams now that are exploring whether it is possible to avoid the uncertainty by introducing gene networks that are embedded in synthetic loops of DNA that are known as plasmids, rather than integrated directly into the chromosomes. And I'll have to make a note of plasmids because I thought I just saw some really strange news about plasmids and some weird crap they found in the ocean. I just copied and pasted that to look into that. And as an extra precaution, their experiments with mice generally make use of engineered cells trapped in implanted capsules rather than by modifying directly the animal's tissue. Well, phew. The field, of course, is still in its infancy. Indeed, the earliest demonstrations of engineering genetic circuits appeared only in early 2000s. But even so, a growing number of scientists grounded in conventional molecular biology are keen to give genetic design a try. Of course they are. Synthetic biologist Ron Weiss, and this would be probably, well, this isn't actually the Weiss with a Y, W-Y-S-S. That is the biologically inspired engineering at Harvard. This is Ron Weiss at MIT. Is teaching an online course on the field that proves its popularity. We have already had about 14,000 people sign up, he says. But the payoff for those entering this field could be huge. I'm in this space, he says, because the frontiers are endless for what biology can do, and it is just a matter of the technology advancing to a point where it opens up those new horizons. Guys, we're at a point where those new horizons are open. We have DARPA investing so much into this from a protection perspective, and we also have them investing in all of these university laboratories. That very guy is giving online courses about the technology. There's no stopping this train. It is coming. It is absolutely coming. I'm going to talk here a little about a couple of the projects that are out for grants. So there is Thor. Thor actually was from 2015. It was archived in September. Technologies for host resilience. So this is out there. There is someone who won it. They are already working on this. This was a DARPA project. Thor strives, and this is TH, little O, big R, technologies for host resilience. I think we are the host. This program strives to discover host tolerance mechanisms in order to identify a whole suite of interventions. Yeah, it means getting it inside of us for use in humans to mitigate the deleterious effects of infectious diseases. And guys, this is how they're going to do it. You know, at one point, Trump came out and he said, oh, vaccines. Yeah, well, that was when that Vax movie came out. He supported that maybe we should back off on the vaccine schedule, but he never, ever came out and said we should back up and look at where these vaccines are coming from. We should back up and take a look at what kind of technologies are now going into these new transgenic vaccines using virus-like particles and all this synthetic biology that we've been talking about here for a long, long time. He never said any of that. And now it's all going to be, since gene editing is a weapon of mass destruction, guys, we have looked at them genetically modifying insects, such as gene drives in mosquitoes. And this Insect Allies program is how entomological warfare was always part of our deep, dark secrets of biological warfare. It never ended. And with this being democratized across the globe, our Department of Defense, big black hole DARPA, has been investing in ways to come up with genetic therapies that could protect us on the fly with synthetic immune systems. That is what this is all about. So in the name of national security, these will be a requirement. And I'm going to tell you, I found something very, 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 very disturbing this morning, because while this election mayhem was going on, Friday, there was a new executive order. And we got about 10, 15 minutes left here. We're going to get to that. Thor will be an intervention to mitigate the horrifying effects of infectious diseases in humans. Thor is seeking novel methods and technologies to discover and understand these responses in hosts, you and me, that are resilient when exposed to infectious diseases. Specifically, DARPA aims to discover the fundamental biological relationships that underlie host tolerance to infection in animal populations, to start with, in order to provide a foundational knowledge to develop interventions for potential transition into clinical use. Step right up to your FEMA camp. You have not been vaccinated, and there has been an outbreak. We have declared martial law, all in the name of keeping you safe. And you now, Johnny, because you are one of the unvaccinated ones, are a threat to national security. This program is going to be organized into 3 technical areas. Number 1, identification and characterization of animal hosts with tolerant phenotypes. So those animals that are easily genetically modified. Number 2, discovery of biological mechanisms that underlie tolerance, meaning those that can be exploited to boost your immunity to something, maybe even on the fly, just like we were talking about with those living factories utilizing different life forms, including mammalian cells, to identify and then produce some sort of prophylaxis. And then number 3, identification of the interventions, that utilize tolerance mechanisms inside a living organism with the goal of reducing morbidity or your death and mortality due to the infection. Yeah, this is going to be the FEMA camp with the new pandemic that will be put into that national stockpile of quote-unquote vaccinations that will be rolled out in a national emergency. The focus of this project will be specifically on understanding tolerance mechanisms in prospective animal studies in conjunction with the analysis of retrospective preclinical and human clinical data sets. Step right up and be our guinea pig for Thor. There's also another program that came out. This was April of 2016 from DARPA. Co-evolving antivirals aim to keep ahead of fast-changing viruses. New program aims to develop adapted therapies able to outpace viral mutations. Zika, Ebola, dengue, influenza, and chikungunya. Ding! We've seen all those. That is all in the collective consciousness. These are but a few among the growing cadre of viruses that today pose serious health threats to U.S. troops, as well as to you, civilian population in the United States and around the world. Vaccines exist for but a few of these infectious diseases, and we've had all kinds of opportunity with these infectious diseases to fund our mad science gone mad in the lab already. And since these viruses have an uncanny ability to mutate and morph, oh yes, as they reproduce inside their hosts, like the never-before-seen Ebola in the eyeballs and the testicles, and the Zika that can be passed sexually transmitted, we've never before seen either. We need vaccine manufacturers, for example, to produce new versions annually at enormous expense. And for that, we have used tobacco also. But ideally, to outpace evolving pathogens, a therapy or vaccine would need to adapt in real time, shape-shifting, ding, 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 as fast as the targets do. And I've talked about the shape-shifting immune system so many times from DARPA. Well, here it is, people. To pursue that radical approach, DARPA launched today, April 2016, its interfering and co-evolving prevention and therapy, more affectionately known as the INTERCEPT program. Viruses are among the simplest infectious entities known, all consisting of just two main parts: genetic material, DNA or RNA, surrounded by a protein shell. He says in preliminary DARPA-funded studies conducted in cell growing in cultured dishes, they have engineered these tips, and we have talked about this. Tips go in and interfere. TIP stands for therapeutic interfering particles, which are tiny virus-like particles. We've talked about this before, that are made of laboratory-grown snippets of genetic material packaged inside protein shells. So just like these viruses, tips can be a Trojan horse, enter host cells, but they have no impact, they say, on their host, but they reproduce like crazy and then starve out the virus that has invaded you. They were talking, I believe this was some of the genetic testing that they were going to look at doing at Penn U at some point in the future, messing around with your T cells. We also found out Microsoft is going to solve cancer in 10 years by reprogramming disease cells. So step right up, people, because here we go. November 4th, 2016. Obama's going to ink it up before he is out of town. Executive order advancing the global health security agenda. Ding, ding. With what? Oh, to achieve a world safe and secure. Really interesting that DARPA just had their Safe Genes program announcement from infectious disease threats. By the authority vested in me as president by the Constitution and laws of the United States of America, it is hereby ordered as followed. As articulated in the National Strategy for Countering Biological Threats and implemented in the Presidential Policy Directive 2. Promoting global health security is a core tenet of our national strategy for countering biological threats. No single nation can be prepared if other nations remain unprepared to counter biological threats, so therefore, it is the policy of the United States to advance the global health security agenda, ding, ding, which is a multifaceted, multi-country initiative intended to accelerate partner countries' measurable capabilities to achieve specific targets to prevent, detect, and respond to infectious disease threats, whether naturally occurring, deliberate, or, accidental. I'm telling you guys, this is just everything we've been talking about, and he just inked it. We are now in charge with these mad science laboratory abominable creations. We have just taken the lead role to vaccinate the planet and take over the human race. I mean, I couldn't even believe this when I read it. The roles, responsibilities, and activities described in this order will support the goals of international health regulations and will be conducted as appropriate in coordination with the World Health Organization, the Food and Agriculture Organization of the United Nations, the World Organization for Animal Health, the Global Partnership Against the Spread of Weapons and Materials of Mass Destruction. Guys, these are all the groups. And what? The International Criminal Police Organization, more affectionately known as Interpol, and other relevant organizations and stakeholders. He's pulling them all together in one big happy group to be able to apply and to forcibly give every single one of us some of this new mad science technology to make us merge with the biopharma tech surveillance. Bag that is closing. All right, guys, I can't even believe we ran out of time. Stick around. Live radio Friday, Saturday, Sunday. Everyone have an awesome weekend, and I will catch up with you on Monday. Everybody take care.
This is JJ